Welcome back, students. This is video lecture on muscles, chapter 9 from the Vander's textbook. This comprehensive chapter is divided into two parts. The first part deals with skeletal muscle. The second part addresses smooth and cardiac muscle. So this tells us the human body has three different types of muscle. Muscles are specialized cells which can generate force, and that force is a mechanical force that pulls on neighboring structures to generate a shortening of some type. Muscles can only shorten, they cannot push and expand. What we have to learn is not just the physiology of each muscle type, but the differences that they have between them. The top row in figure 9.1 shows actual pictures taken with the light microscope of various layers of muscles of the three types. So here we have skeletal muscle, and one can see the tiny little striations responsible for its characteristic appearance. The other thing is that the cells are multinucleated, so they are fused together to form giant cells. In the case of smooth muscle cells are arranged in a spindle type of arrangement with normally a single nucleus embedded inside the cell. There are no striations because, as we'll find out later, there's no absolute organization. And in the middle we have cardiac muscles. Cardiac muscles resemble both types, the smooth and striated. With regard to the striated, they have striations too. That tells them that they have internal organization, very similar to skeletal muscle. But they are, once again, single nucleated, spindle-shaped cells that are attached to each other through these intercalated discs. One other comment about individual cardiac cells is that they are branched, so they make contact with numerous neighboring cells. The majority of this chapter focuses on skeletal muscle. Indeed, learning the physiology of skeletal muscle allows us to set the baseline, and then we can simply look at the differences in smooth and cardiac cells. The terminology can be confusing, but the baseline is to consider the muscle cell itself. A muscle cell is known as a muscle fiber. These cells have certain characteristics which help them in their physiology. The first thing is that they are multinucleated. Not many cells in the human body are multinucleated, but striated muscle cells are one class which are. And that's to do with their developmental processes which lead from stem cells to giant muscle cells of this nature. The second thing is they contain many mitochondria. Mitochondria generate lots of ATP, so these cell types require a substantial amount of ATP energy for their functionality. In fact, we'll learn that ATP is required in 4 different processes when it comes to the activity of these cells. Third, they have an unusual internal structure to their cells in that they have transverse tubules. Transverse tubules are nothing more than the plasma membrane of the cell diving inwards to form invaginations that then penetrate deep into these cells. We'll see the functionality of this a little later. Fourth, they have myofibrils and sarcomeres. So these are the internal arrangements of proteins that give them a unique appearance. And the sarcomere is the functional unit of a skeletal muscle cell. Lastly, but unusually, these cells have the same structures as any other cell, except that the names have been supplemented. So whenever we talk about the sarcolemma, we're talking about the plasma membrane. When we are talking about the sarcoplasm, we're talking about the cytoplasm. And when we're talking about the sarcoplasmic reticulum, we're talking about the endoplasmic reticulum. The smooth endoplasmic reticulum in this case. So basically, we're using the word sarc to indicate that these are skeletal muscle cells. Figure 9.2 nicely puts the anatomical nature of the hierarchy of muscle cells into perspective. The wedge that I have drawn on the left-hand side of the slide helps me remember the hierarchy. So there are 2 elements on the left side of the wedge and 2 on the right, with a pinnacle being the muscle cell itself. The muscle fiber is this indicated here. You can see 2 of the nuclei attached to its extreme periphery, but most of the cell will be full of proteins arranged in this very, very orderly fashion, with these striations evident from one angle. If you go above that, you will end up with the muscle bundle. A muscle bundle is simply a collection of muscle cells attached to each other through connective tissue. And then when you have enough of these muscle bundles, you end up with the muscle itself, which can have significant proportions, such as the muscles in your back and those in your legs. Progressing down the other side of the wedge, we go into the cell. So the myofibril is this protein construction consisting of actin and myosin and a number of other proteins. And these can litter the inside of a cell in various quantities. And then finally, along the length of the myofibril, we have regions, which are identified as the functional units of the muscle cells. And here we have an exaggerated view of a sarcomere, and these sarcomeres simply repeat one after the other, sometimes tens of thousands of times per myofibril. And we'll get into the details of the functionality and the anatomy of sarcomeres shortly. The myofibrils, the internal structures consisting of protein, they are normally 1 to 2 micrometers in diameter, and that size doesn't change much. However, their length can vary, and sometimes it can go from one side of a muscle fiber to the other, and this can be enormous distance measured in centimeters rather than micrometers. One needs to remember that the thin filaments are made of actin, and the thick filaments are made of myosin. These two may represent the major proteins present inside skeletal muscle, but by no means the only proteins present. Other proteins that we will have to consider are titin, tropomyosin, troponin, and a few others that are involved in the structures of the sarcomere. At the molecular level of detail that we need to understand, we can look at myosin and actin as two separate entities which have internal organization. Let's begin by looking at the thick filaments. As we said earlier, they consist mainly of the protein myosin, not exclusively. The myosin molecule is composed of two large polypeptide heavy chains which are twisted around each other to form a helix, and then at the ends they have globular domains, and those globular proteins play an important role in binding to either actin or to a molecule of ATP. If we revisit figure 9.3, we can see that these are the heavy chains in light brown, and the light chains are here in slightly dark shades. The heads have two binding sites, one for actin at its tip and the other for ATP. This particular region of the myosin protein heavy chain also has an enzymatic function called the myosin ATPase. This domain is able to break up ATP into ADP. The thin filaments have a more compound nature. The heart consists of globular actin subunits, and these subunits are attached to each other to form very long fibers. Associated with the actin is another newly discovered protein called nebulin, which is thought to have a role in assembly of these filaments. In addition, two other types of proteins sit on the surface of the actin, and those proteins are known as troponin and tropomyosin. So we go back to this figure here, we can see that the troponin is in pink, and the tropomyosin is a long filamentous protein that occupies a groove along the surface of the actin. The precise interaction between actin, tropomyosin, and troponin is very important. If one looks closely, one can see little tiny dots on the surface of each of the actin subunits. And those black dots indicate a binding site for the surface of the myosin protein. Under normal circumstances, when the muscle is not contracting, the tropomyosin is covering these binding sites so that there is no interaction between the two types of major protein. However, under certain circumstances, the tropomyosin can be shifted away from these locations, therefore revealing the binding sites for myosin and then allowing something called a cross-bridge cycle, which we'll learn about a little later, to take place. So a very dynamic structure exists on the surface of the actin, and also with regard to the myosin that we'll discover later. Figure 9.4 is very important for students to relate to, both at the static level and at the dynamic level when things are contracting. One thing to understand is that the sarcomere or the sarcomere has limits. So the Z lines are the limits of each sarcomere. So this is one sarcomere, and then this will be the beginning of another on the right, and yet another one on the left. And this will repeat multiple times along the length of the microfibril. There's a whole section coming up that deals with this. The angle of view of muscle fibers is very important. If one looks at it from the side, we can see the sarcomeres and the striations. If one looks at it in cross-section, we can see that there are specific arrangements of the thin and thick filaments. The thick filaments are in brown and the thin filaments are in purple. In order to fully grasp the functionality of a striated muscle cell, the overall interaction between different components of the cell is fundamental to this understanding. Students are highly encouraged to spend a few minutes understanding the architecture presented by figure 9.6. Of particular note is the orientation of the plasma membrane, shown here in light brown. These little tubes that develop at the surface, they dive deep into the heart of the cell, but they're still part of the plasma membrane. And these are what we call the transverse tubules. Their purpose is to take any electrical activity that materializes along the surface of this cell and help it penetrate deep inside without delay. Next, notice the close association between the sarcoplasmic reticulum and the T-tubules. There must be some need to position these so close together, and we'll see that in the future. The sarcoplasmic reticulum is quite extensive, and it permeates throughout the entire cell as close as possible to each myofibril. Finally, notice that the mitochondria are positioned to be in close proximity to supply huge amounts of ATP to an active muscle cell. In addition, the arrangement of the sarcoplasmic reticulum isn't uniform throughout the cell. As one approaches each end, we have large regions of blue here, and those are known as the terminal cisternae, and they are designed to hold large amounts of something. And what they hold is calcium. So large amounts of calcium is bound to proteins inside these terminal cisternae, and the name of that protein is calsequestrin. So calsequestrin stands for calcium sequestering protein. And this gives us a clue as to how the activity of the skeletal muscle cells can be regulated, and it has to do with the amount of calcium present inside the cytosol. The T-tubules lies directly between and in close association with the terminal cisternae, as we saw in the previous figure. And finally, the T-tubules are continuous with the cell membrane, the plasma membrane, also known as the sarcolemma. And this arrangement is key in dictating the rate, the speed at which the chemistry can activate these cells. Just to reiterate and to warn students not to forget certain things, the term contraction, when applied to muscle cells, does not necessarily mean shortening. It refers to a physical force that's generated by these cells using the cross-bridge method of generating force, and that force can be used to maintain a status quo. For instance, somebody standing in the gym holding a dumbbell, which is akin to isometric exercise. Regardless, the muscle is still undergoing chemical changes in order to generate the force. Once the nerve impulses to the muscle are extinguished, turned off, the tension declines, and then the muscle is able to relax. Another area of debate among students is the use of the word motor unit. A motor unit is defined as the motor neuron, a nerve, and the associated muscle cells that it innervates. With respect to skeletal muscles, a single motor neuron can innervate many muscle fibers, but each muscle fiber is controlled by a branch from only one motor neuron. So all for one and one for all. An alternative explanation is that a single nerve fiber, when activated by the central nervous system, will drive the contraction of a number of muscle cells. It's an all or nothing. So either all the muscle cells are activated or none are. So that will be a motor unit. A whole muscle consists of many motor units, which can be activated according to both a learned behavior and an innate behavior. Figure 9.7 demonstrates this. In the top half, we have a single neuron activating 5 different muscle cells. And in the bottom diagram, we have a single neuron in purple activating 4 muscle cells, and a second neuron in orange activating 3 muscle cells. So you could either fire one or the other, or both, or neither. Under normal physiological conditions, serrated muscle has to be activated using motor neurons. The point at which the nerve makes contact with the surface of the muscle cell is known as a neuromuscular junction. By anatomical design, motor neurons always have their cell bodies located either in the brainstem or in the spinal cord. But that's a subject of another chapter. For now, we need to understand that the speed at which these signals travel down these nerves can be affected both by the diameter of the nerve. The larger the diameter, the faster the chemistry moves, as well as by support from neighboring cells in a process known as myelination. And we'll see that in a different chapter. Figure 9.8a shows beautifully these neuromuscular junctions at the intersection of motor neurons and muscle fibers. Understanding the physiology of a neuromuscular junction is one of the major goals of this chapter. So students are encouraged to recall exactly what happens as a sequence of events at a neuromuscular junction. The action potential travels down the axon of the motor neuron, and when it hits the buton, the nerve end, it then translates itself into the release of calcium into the bulge, and that calcium encourages the fusion of these vesicles that contain neurotransmitter with the end plate membrane of the neuron cell. And that fusion releases into the cleft a neurotransmitter. And that neurotransmitter then travels across the cleft and then interacts with receptors on the surface of the muscle cell. In all cases of motor neurons, the neurotransmitter in these vesicles is acetylcholine. Once the acetylcholine binds to its receptor, it causes the influx, the movement of huge amounts of sodium across into the cell. And that creates a membrane potential, and that membrane potential quickly runs in all directions away from the motor plate. All neuromuscular junctions contain neurotransmitters which excite the neighboring cell, in this case the muscle cell. However, to minimize the duration of the activation, there are various mechanisms available to quickly mop up the neurotransmitter. And one mechanism is to use an enzyme, acetylcholinesterase, that rapidly breaks down the neurotransmitter. We have learnt a lot about how this particular junction functions by looking at various diseases and situations. One of the most curious is the arrowhead poison used by indigenous tribes in South America. So this normally comes from the skin of a frog, and they rub their arrowheads onto the skin of the frog, therefore transferring the poison. to be used on their victims. The active ingredient is curare, and that binds strongly to acetylcholine receptors, but it doesn't trigger the muscle cell in generating an action potential. So basically it binds and blocks for long periods of time so that no natural acetylcholine can activate the muscle cells. And that leads to the breathing muscles becoming paralyzed and the victim dying of asphyxiation. Other agents work in alternative mechanisms. One of these is the organophosphates, which are sometimes found in pesticides, and also nerve gases. What they tend to do is they inhibit the enzyme acetylcholinesterase used for destroying acetylcholine. The ultimate result is that the breathing apparatus is paralyzed, and once again the victim dies of asphyxiation. The final example illustrates the functionality of Botox. Botox is a toxin produced by the bacterium Clostridium botulinum, which works under natural conditions by preventing the vesicles from fusing with the membrane of the nerve cell. And that relies on the activity of snare proteins within that cytoplasm. Moving on to another principle of muscle activity, that of the relationship between the action potential arriving at the surface of the plasma membrane of the muscle cell and the resulting contraction that's realized as the activity of the contractile apparatus. In these charts, it's important to keep an eye on the axis. In this example, the axis is time in milliseconds, and then we have two different y-axes for each of these graphs. The top one is basically telling us the membrane potential developed on the surface of the muscle cell. You can see that the muscle cell is normally minus 90 millivolts internal, and it can become positive 30 millivolts internal due to nerve activity. And that is called an action potential. In response, the interior of that same cell will generate a force, and that's measured in milligrams. That's the muscle fiber tension. And you can see there's a small delay called the latent period between the action potential propagating around the cell and down into the T-tubules before the sarcomeres begin to contract and generate force. That latent period is due to the molecular biology of the cell in activating and releasing calcium, and then that calcium causing the tropomyosin to do its movement. The relative slowness of the muscle contraction is then illustrated by the depth over which this activity takes place. The cross-bridge cycle is best understood by watching an animation of the entire process. Regardless, we'll describe the events right here. Under normal circumstances, when a muscle cell is not activated and it's relaxed, the situation at each sarcomere is as follows. The myosin heads are not in contact with the actin due to the presence of tropomyosin, which covers and thereby prevents interaction between the actin and the myosin. The ultimate location of the tropomyosin is under the direct control of troponin. So troponin is the master of tropomyosin. The troponin itself is composed of 3 subunits of protein designated as I, inhibitory, T for tropomyosin binding, and C for calcium binding. And the last one gives you a clue as to how the system can be activated. In the presence of calcium, which is released due to nerve activity, the calcium binds to one of the subunits of troponin, causing a knock-on effect onto the other subunits of troponin, which results in a repositioning of the tropomyosin, revealing for the first time the heads on the actin, the myosin binding heads. And that allows the formation of these cross-bridge cycles, which then activates a walking behavior known as the cross-bridge cycle, and we'll look at that later. Regardless, at that point, force is generated within the muscle fiber. Subsequently, the calcium is mopped back up again, and the system reverses in that the calcium binding subunit repositions the troponin and the tropomyosin to recover the heads, resulting in the eventual release of the myosin from the actin. The connection between the plasma membrane, the surface of the muscle cell, and the mechanism that leads to the cross-bridge cycle is actually a little more complex. Figure 9.12 reveals this complexity by showing this insert. At the insert point, we have a close relationship between a cell membrane receptor and a receptor encoded in the membrane of the sarcoplasmic reticulum. So these two membranes are closely opposed, and the receptors are actually touching one another. This translates to the following. As the action potential travels along the surface of the plasma membrane of the muscle cell, it eventually reaches these modified voltage-gated calcium channels, which are officially known by the name of dihydropyridine, DHP. As the DHP channels open, they physically alter their configuration, and that physical change initiates an opening of the calcium ion channels, which are located on the sarcoplasmic reticulum. These two have a scientific label, and they're called ryanodine receptors. That allows huge amounts of calcium stored in the cisternae of the sarcoplasmic reticulum to then enter the cytosol, the yellow space of the muscle cell. And that triggers the cross-bridge cycle, which is part of the sliding filament mechanism, which allows the shortening of muscle cells. The mechanism makes full sense once somebody looks at the molecular detail of how the proteins within the sarcomere slide past each other. So once again, the myosin is represented by the large brown structure, and the actin is represented by these thinner structures. And during the cross-bridge cycle, the contact between the two types of protein allows the actin to move in, sliding past the myosin filaments. That's indicated here in a bit more detail. So just looking at one sarcomere from this point to this point, these will be the Z lines. And we can see in a relaxed muscle, the distance between the Z lines is quite significant. But in a shortened muscle, the Z lines come together, therefore reducing the distance between them. And that repeats again and again with every single sarcomere, shortening the muscle. Students are highly encouraged to watch animations showing the sliding filament hypothesis, as well as learning the names of each of these zones and bands and protein locations, because they explain nicely what's happening. The H zone corresponds to just the region of the sarcomere in the middle, which is composed of myosin, not including the actin. The I band does the same thing about the Z line, so just the myosin at the location of the Z line. The A band, however, corresponds to the distance that the myosin occupies, as well as its overlap with actin. During shortening, the A band does not change because the length of the myosin filaments is static. However, the amount of overlap between the actin and the myosin reduces the size of the I band and the H zone in each sarcomere. The myosin filaments are held together in place by another protein located right here, and that corresponds to the M line, M standing for middle. During muscle relaxation, the entire process reverses and the sarcomeres are pulled back out by various other forces. Figure 9.15 deals with the famous cross-bridge cycle that we've been alluding to. This cycle illustrates a sequence of events which take place when calcium is released into the cytosol of muscle cells. The rudimentary aspects of this cycle are illustrated by the following video.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Figure 9.15 provides a little more depth to this animation.
A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross-bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum.
The involvement of ATP in muscle contraction cannot be overstated. It powers various components allowing muscle contraction to ensue. The first place that ATP hydrolysis is very important is maintaining the gradient on the surface of the muscle cell. Without ATP hydrolysis, the sodium-potassium ATPase, or the sodium-potassium pump, is unable to maintain the gradients of sodium and potassium necessary for the action potential to materialize. The second location involving the use of ATP is by the calcium ATPase. These proteins are active transport proteins embedded in the sarcoplasmic reticulum. and under appropriate signals, they are able to move huge amounts of calcium back into the reticulum when the action potential has decayed, thereby lowering the calcium ion concentration in the cytosol, ending the contraction of the cross-bridge cycle. And thirdly, as we just saw, the hydrolysis of ATP by the myosin ATPase, the cocked heads of the myosin, is necessary in order to walk along the actin filament in the cross-bridge cycle. The next two slides detail step by step the sequence of events between a motor neuron action potential arriving at the buton and the skeletal muscle fiber contraction and relaxation. Students are highly encouraged to relate to these sequence of events and to get them in the correct order. Let's turn our focus now to the mechanics of muscle fiber contraction. The first thing to understand is the word tension. Tension is a force, a physical force exerted by tissue on its surroundings. And in the reciprocal sense, the load is the force exerted on the muscle by the object. Thus, these two forces, tension and load, are opposing forces. If one is larger than the other, then either something is moved or it's not moved. The minimum response of a muscle cell to a single action potential is known as a twitch. Physiological investigation of twitches has revealed a lot about the mechanisms of operation of various body muscles. The purpose of figure 9.16 is to illustrate the difference between isometric and isotonic muscle twitching. In each of these experimental setups, a single muscle is activated by a single motor neuron, and the device can either measure the load that the muscle fiber is able to lift or the force exerted on a static plate. In the upper experiment, the muscle fiber is attached to an immovable transduction plate. When an action potential is fired down the motor neuron, there's a small delay called the latent period before a tension is recorded in the muscle fiber. So here we have a tension profile. Because the muscle cannot move the plate because it's fixed, this will be an isometric twitch recording. Isometric means that the muscle fiber, even though it doesn't shorten, does generate a force on the objects around it, in this case, the instrument. The purpose of experiments such as these in isometric contraction is to show the profile of the forces generated within the fiber. So over a period of about 30 to 35 milliseconds, the force builds up as the cross-bridge cycles are activated, and then after that period, the force decays as the calcium is removed from the cytosol and the number of cross-bridge cycles is reduced. The lower experiment reveals that a muscle fiber can lift a load, and if the load is within its range, it will be lifted, but the latent period is increased as the muscle fiber overcomes the load itself. So depending on the load, the muscle fiber may have a longer or shorter latent period. But the profile here represents the effects of this chemistry on the mechanics of the muscle. For any muscle lifting a load, there are three segments to that isotonic twitch. The first is the latent period. That's the force generating stage to overcome the load. Then we have the contraction time. That's the time interval from the beginning of tension development at the end of the latent period to the peak tension development. So in this case, that would be this time period here. And then finally, we have the relaxation time, and that's the time interval from peak tension until the tension goes back to zero. And that, in corresponding to this, will be from this point to this point. So that'll be that time period. This all depends on the muscle fibers and also depends on the load. During all muscle contractions, first there's isometric contraction. That's the period where the tension is developing in the muscle, but the muscle cannot shorten because the load has to be overcome. Isometric training exercises are important in that in those instances the individual is pressing against something that's not movable, but they are still exercising their muscles by generating this tension. If and when the tension in the muscle overcomes the load, then we transition to isotonic contraction condition. The relationship between isotonic twitches and different types of load for any single muscle fiber is indicated by this chart here. So the action potential reaches the muscle at time 0. There's a latent period, and the latent period can vary depending on the load. So we have a light load in green and a very heavy load in purple, and we can see a medium load in orange. So initially, when the internal forces are overcome and the muscle generates enough tension to start moving the load will be moved based on a profile given here, and that is called the shortening velocity, a very important concept. So the angle here, the slope of this line on this side, is the shortening velocity. So the steeper this line, this dotted line, the greater the shortening velocity. So over a short period of time, that muscle fiber will contract, lifting the load. If the load is heavy, the first thing to notice, it takes a longer period of time for the internal forces, the tension in the muscle, to overcome the load. And once the load starts moving, the slope indicates that the shortening velocity is quite shallow. Therefore, the load is not moved a significant distance because of its mass. If you relate this to exercising in the gym, for instance, lifting dumbbells, a light load can be lifted quite easily and quite quickly, whereas a heavy load takes a longer period to overcome, and then it can't be lifted as easily by the same muscle. The kindest way to illustrate the concept here is to take you back to the gym. Let's pick a point. Let's pick this point right here. So maximum isometric tension. So this will be a condition where you're pushing a barbell containing a weight, for instance, doing a bench press. And here the weight is not moving. You're pushing as hard as you can with your chest muscles, but the weight has come to a standstill. The shortening velocity is therefore 0, and the load is the maximum that that muscle can carry. Anything below the 0 point would mean that you are losing control of the weight, and even though you are pushing, your muscles in your chest are beginning to lengthen. Conversely, the other side, the lighter the load that you're bench pressing, the easier it's to move up and down. So a very light load you can push very quickly back and forth, and your shortening velocity is the maximum for that chest muscle. Whereas down here, you're experiencing a greater and greater load, therefore the shortening velocity has decreased. In the experimental analysis that we did a few slides back, we're looking at a single muscle cell and a single action potential arriving at the surface of that muscle cell. Now, we did notice that the action potential only lasts 1 or 2 milliseconds, whereas the twitch that's generated in that muscle can last for 100 milliseconds or even longer. What would happen if a second action potential arrived down the nerve before the mechanical contraction of the muscle tension had died away? That is, before the first muscle twitch had subsided. This is exactly what figure 9.19 wants us to understand. So let's go to the first panel here. So we have time on the x-axis and we have muscle tension generated in that fiber on the y-axis. A single action potential will allow the tension in the muscle to have the traditional profile that we looked at earlier. If you have a second action potential which originates after the tension in the muscle fiber has died away, then you get an exactly similar profile generating after that time period. In this case, you get the first action potential, and before the decay of the tension in the muscle, you have a second action potential following close by. So you have a bip bip. And here you can see that the muscle is caused to generate even more tension than with a single individual action potential. And if these are put together even closer, then you get a summation effect where the activity, the tension in the muscle becomes added, and you get a greater profile. So this looks like this, but to a greater tension level. As the action potential stimulus is increased in frequency, eventually you'll get to a profile where the muscle does not have any undulations. So here the frequency of activation is so far apart that the muscle has time to respond by producing individual waves of tension. But here the nerve may be stimulating the muscle at a very rapid rate, and the muscle will then enter something called fused tetanus. Fused tetanus. Whereas this segment here is called unfused tetanus. The reason we use the word tetanus is that the muscle always has tension in it. Tetanus means tension. And you can see during this period here, the tension never returns back to normal because there's always a follow-up stimulus causing the muscle to fire again. In this case, there are no undulations, and this is called a fused tetanus. Students need to be familiar with both these terms. Here the stimulus factor is 10 stimuli per second, and here we have 100 stimuli per second. Lastly, let's look at the LT relationship, the length-tension relationship. Another analogy may be useful in understanding this concept. Going back to the gym, when we're exercising our biceps, for instance, we tend to move the barbell between 2 points, but we never start normally with a fully stretched arm, as in the case of preacher curls, because the load that we can lift is not as great as we would if we were just bent around the elbow. And conversely, we never do bench presses with the arm fully contracted. Thus, the maximum force that our bicep can generate is when it is not fully extended or fully contracted. And that can be related back to this figure here. So this is the length of the muscle, the bicep, and we can see that it's very inefficient at generating force when it's fully contracted or it's fully extended. And there's an optimum area here where the bicep will have the maximum force generating ability, and that's called the optimum length of that muscle. This relates back to the overlap between the actin and the myosin filaments, in addition to the recoil generated by a protein called titin, which anchors the sarcomere to the plasma membrane of the muscle cell. The next section looks at the biochemical properties of different types of muscle fiber. So in the body, we have various types of muscle cell that can be used in various proportions to make a muscle. The first of these parameters is the way that energy is extracted from food, the primary purpose of which is to make ATP to drive all the processes that we learned a little earlier. Regeneration of ATP can occur by three mechanisms. The first is phosphorylation of ADP by a substance called creatine phosphate. The second is through mitochondria with oxidative phosphorylation that involves oxygen and the electron transport chain. And then finally, we can have phosphorylation of ADP by glycolysis in the cytosol. That doesn't have the requirement for oxygen. And the byproduct here would be pyruvate. The byproduct here would be water and carbon dioxide. The ratio of these three sources of ATP production in muscle cells can vary, and that gives rise to the different types of muscle fibers. A second parameter we have to take into consideration before looking at the types of muscle fiber is muscle fatigue. Muscle fatigue is described or defined as the inability of a muscle to continue to keep responding to a stimulus in generating tension. So we all know that muscles fatigue over time. Some muscles fatigue earlier than others, and that's what we're talking about. So muscle fatigue is a physiological term referring to the decline in muscle tension. A consequence of muscle fatigue is a decrease in the shortening velocity, and also a slower rate of relaxation. Fatigue is not the same for every single type of muscle in the body, so the onset of fatigue and its rate of development depend on three parameters. The type of skeletal muscle fiber that is active, I'm going to look at that shortly, the intensity and duration of the contractile activity, how long and how intense, and the degree of one's fitness. Returning back to the gym as an example, we know that the first time we pump a piece of iron, we can do it quite readily and maintain it. But with every subsequent cycle, our ability to repeat that decays. So we have muscle fatigue setting in. So tetanus cannot be maintained for as long through each repeated cycle. The full reason for muscle fatigue is not yet understood well, but we do have some indicators, and many factors can contribute to muscle fatigue, and some of them include a decrease in the production of ATP from ADP, an increase in the concentrations of waste products, and free radicals. So magnesium ions are produced as a result of various biochemical maneuvers, and so is pH acid. These biochemical outcomes have been correlated directly with the cell biology. So whenever you have either a decrease in ATP or an increase in these products, there's a decrease in the rate at which calcium can be recycled within the cell, its release, its reuptake, and storage. There's also a decrease in the sensitivity of the thin filament proteins to activation by calcium, and also the cross-bridge cycles themselves may be inhibited, and the power that they generate may be interfered with by these substances. Muscle fatigue can be caused by two locations. One is your central nervous system, where your will to do something has faded, and that's willpower. And the second is the muscle itself, the biochemistry of the muscle, as we just saw in the previous slide. Putting all the pieces together, we arrive at the conclusion that our bodies contain three basic types of muscle fiber. The two important parameters are the speed at which the shortening takes place, the velocities of shortening. Some muscles can shorten faster, others slower. And the second is the way that they utilize energy to make ATP. The underlying cause for reason one, these velocities of shortening, appears to be that the body can manufacture myosin in different subtypes. And those subtypes then interact with the chemistry around them, therefore either generating fast or slow twitch fibers. The second parameter can be explained by looking at the biochemical complement of these muscle cells. So some fibers contain many mitochondria, and that allows them to have a very high capacity for oxidative phosphorylation. And those fibers are known as oxidative fibers, OFs. These OF fibers contain a large quantity of a protein called myoglobin, and that makes the fibers appear darker. Thus, these are sometimes known as red muscle fibers. Because of their enhanced capacity to produce lots of ATP using oxygen and fuel, they are utilized for maintaining posture, so long-term contractions, such as the muscles in your back. The other class of muscle fiber cells are called white muscle fibers because they do lack this myoglobin protein. These fibers are equipped with a slightly different complement of biochemical mechanisms, and they are known as glycolytic fibers. And they don't have as many mitochondria. They do have a large complement of enzymes involved in glycolysis, and they can store significant quantities of glycogen as a source of glucose backup. So this gives these muscle fibers the ability to produce quick bursts of activity, but they will fatigue very quickly. Thus, the three main types of muscle fibers are listed here. So we have the type 1, which are the slow responding, and they use oxidative biochemistry. We have the type 2A, which are the fast responding, and they use a combination of oxidative and glycolytic biochemistry. And then we have the type 2X, which are the fast responding, and they're the glycolytic type. And depending on the needs of that particular muscle, the combination of these, the ratio of these, can be appropriately adjusted. Not only that, through exercise and other means, we can change the ratio over a period of time. Figure 924 shows a typical skeletal muscle cross-section, and by using different staining techniques, we're able to identify the three types of fibers. So the blue cells are type 1 fibers, the green cells are type 2A, and then the black cells, or the nearly black cells, are the 2X types. Figure 925 graphically illustrates the fatigue rate of the three types of fibers. So the first type of fiber is the slow oxidative fibers, and they can maintain a tension over a long period of time. The second type are the fast oxidative glycolytic fibers, and they do fatigue but slowly over time. And then lastly are the fast glycolytic fibers, which do fatigue rapidly, and these are used only for short bursts of activity. Table 9.3 does a great job of comparing and contrasting the three types of muscle fiber found in human beings. Students are highly encouraged to spend some time looking over and learning the characteristics of the various muscle fibers. Figure 9.26 relays the association between motor units and the type of muscle fiber. So in this particular example, motor unit 1 will be impinging on these four slow oxidative fibers. Motor unit 2 will be activating the fast oxidative glycolytic fibers in orange. And motor unit 3 would be innervating the fast glycolytic fibers. Initially, the muscle may only be activated using motor unit 1, but if that's not enough to lift a load, so the tension built here is not enough to lift the load, then after a short period of time, the brain may decide to activate motor unit 2. And if that in compound relationship with motor unit 1 is not enough to lift the load, then a third motor unit may be activated. And hopefully that will lift the load. If not, more motor units could be recruited by the central nervous system. This slide relays the conclusions we have reached so far, that depending on the needs of the body, the proportions of fiber types present in different muscles can be altered. So the back muscles, which need to do long-term fatigue-free work, would be composed of large numbers of slow oxidative fibers, but not exclusively. And of course, the muscles in the arms, which are normally used for quick responses, would have a large proportion of fast glycolytic fibers. And the leg muscles, used for fast running over intermediate distances, would have a high proportion of fast oxidative glycolytic fibers. So overall, we've covered all three types of muscle fiber in this set of examples. In concluding this section of the chapter, the next few slides allow us to make some general conclusions about muscle tension. So the total tension that a muscle can develop depends on two factors: the amount of tension developed by each fiber, each muscle cell, and the total number of fibers that are contracting at any one time. So by adjusting between these two, the body can control whole muscle tension as well as the shortening velocity of that muscle. Which factors determine muscle tension? Well, we now know that tension is developed by each muscle fiber based on the action potential received, the frequency of activation by motor neurons, the length of the fiber, the diameter of the fiber, the type of the fiber, and of course the rate of fatigue. Combine the above with the number of fibers per motor unit and the number of active motor units, you can see that we have whole cell control to a very large degree over that muscle. The other discovery in physiology is that the ratio of motor units to nerves can vary depending on the muscle. Some muscles have fine control. That is, the number of nerves innervating muscles, the ratio is pretty low. In other areas, a single nerve can innervate thousands and thousands of muscle cells. Therefore, that gives you more coarse control over that muscle. So the eyes have muscles, and those eye muscles are under very fine control, as are the muscles in your fingertips. But the muscles in your back may be under coarse control. So the ratio can vary from one area to another. If one exercises muscles, the muscles will increase in the number of muscle cells that they have inside that muscle. And they can also change the capacity of the ATP production and the subtype of myosin that they express over time. So one can exercise more efficiency to align with the type of activity one is doing. On the converse side, if you don't use your muscles, then they will atrophy, either as a result of non-use, called disuse atrophy. And the example they gave is you put your arm in a cast over a long period of time. And the second is nerve damage that can lead to muscle atrophy. As we alluded to at the very beginning of this video lecture, muscles can only contract. They cannot lengthen using force. Evolution has thus generated antagonistic muscle groups. So the bicep is used for flexing your arm, and the tricep is used for extending your arm. And all over the body, you have these antagonistic muscles acting against each other. Simple physical principles can be applied to the way that muscles exert their force, their tension. In this example, the bicep acts as a fulcrum, as do many locations throughout the body in conjunction with the skeleton and the use of tendons. The textbook next discusses a number of different muscle disorders, beginning with polio and how polio causes a destruction in the motor neurons themselves, leading to respiratory failure. Then we look at muscle cramping, which is not fully understood, but we believe here we have electrolyte imbalances and dehydration responsible for this. Then diseases related to the concentration of calcium in the extracellular fluid. So hypocalcemic tetany is an example here. Even though we don't fully understand how that works, there is a relationship between extracellular calcium levels and intracellular functioning of the cross-bridge cycles. The next slide discusses muscular dystrophy, including Duchenne muscular dystrophy, and this is quite common in males and has something to do with the way that the force-generating components are connected to the plasma membrane of the muscle fibers. And those regions are called costomeres. The second part of the chapter addresses smooth muscles and cardiac muscles. Smooth muscle cells have some similarity to skeletal muscles in that they contain actin and myosin filaments, but they don't have any troponin. Therefore, one can predict that their force generation mechanism may be slightly different. And that's true. We'll see that shortly. The other major differences are that these cells, the smooth muscle cells, are normally spindle-shaped, and they have a typical cell diameter ranging from 2 to 10 micrometers, and their length can be quite exaggerated, up to 400 micrometers. But they're nowhere as long as skeletal muscles. Smooth muscle cells have a single nucleus, and they also have a continued capacity to divide throughout their lives, depending on the needs of that local tissue. Finally, there are some special proteins called dense bodies, which allow the thin filaments to be anchored to the plasma membrane, therefore transmitting the mechanical force. The internal cellular organization of smooth muscle cells does not give rise to myofibrils. Therefore, there are no sarcomeres within these cells. And that accounts for the absence of the striations that are responsible for discriminating smooth cells from both cardiac and skeletal muscles. However, the force generated within smooth muscle does occur by a form of sliding filament mechanism. And we'll look at that on the next slide. Smooth muscle cells surround hollow structures and organs that undergo changes in volume, like the spleen. And they accompany these changes by contracting and reducing the lumen of that particular space. So around blood vessels, smooth muscle cells are present in concentric rings, and when they contract, they cause the diameter of the blood vessel to be reduced. Figure 9.3.3 shows a relaxed smooth muscle cell as well as a contracted smooth muscle cell in a diagrammatic form. And the insert shows us the arrangement of thick and thin filaments. The thin filaments are anchored onto the dense bodies, whereas the thick filaments are hovering in between the thin filaments. Smooth muscle cells do undergo cross-bridge cycling in a manner very similar to the ones that we saw in striated muscles. However, the trigger that initiates this process is different. It doesn't require troponin or tropomyosin. In place, it uses an enzyme, and that enzyme is regulated by the concentration of calcium within the cytosol. And as the level of the calcium increases, the activity of the enzyme increases, and that phosphorylates by adding phosphate groups to certain amino acids on the myosin. And just the phosphorylated form of myosin can participate in cross-bridge cycling. So controlling phosphorylation and dephosphorylation is the key to activation and deactivation of smooth muscle activity. The sequence of events leads to the activation of myosin and not to the activation of the thin filaments in actin, which is different to skeletal muscle. So the process begins with an increase in the concentration of cytosolic calcium, and that binds to a protein called calmodulin. Calmodulin is present in all cells, not just smooth muscle cells. The reformed calmodulin calcium complex is then able to activate a second protein called myosin light chain kinase, and the combined action of these two entities results in an active form of this protein, and that can then pick up ATP from the cytosol and add that to the light chains of the myosin. And by adding ATP to the light chains of the myosin, that phosphorylates those light chains and releases ADP. And the phosphorylated light chains have a knock-on effect in that they cause the globular domains of the myosin thick filaments to be extended out towards the actin binding sites on the actin filaments. This will continue cycling as long as the cytosolic levels of calcium are high and calmodulin is activating the kinase. The reverse takes place when levels of calcium drop. Another protein called myosin light chain phosphatase then removes the phosphate groups, restoring the myosin to the closed configuration. The comparison of smooth muscle activity with skeletal muscles is presented in figure 9.35, whereas skeletal muscles are always activated by motor neurons. Smooth muscle activity can be influenced by various alternative types of input. One is the electrical activity on the surface of these cells, and that can happen spontaneously due to local conditions. Neurotransmitters released by the autonomic nervous system can also activate the smooth muscle cells, as can hormones traveling through the blood, and local changes such as levels of pH and oxygen and ion concentrations. And finally, a mechanical stretch of these cells in some cases can trigger them to contract. The first type is very important, the spontaneous electrical activity at the surface of the plasma membrane. A few types of smooth muscle cells can generate action potentials themselves spontaneously, and they don't require any input from any other system beyond. And an example of these will be the pacemaker potential cells that we are familiar with in some parts of the body. A good example being the length of the GI tract, which has these pacemaker cells that can rhythmically contract the smooth muscles, leading to slow peristalsis or segmentation. These smooth muscles are innervated not by the motor neurons, but by the autonomic nervous system. Each nerve fiber of the autonomic branch has buttons or growths called varicosities, and these varicosities are the points at which neurotransmitter is released onto the surface of the smooth muscle cells. And each nerve fiber can have multiple varicosities, therefore make contact with numerous smooth muscle cells. In some circumstances, a single muscle cell may be located close to a number of these varicosities belonging to different classes of nerve cells, both belonging to the sympathetic branch of the nervous system and the parasympathetic branch of the nervous system. With respect to the interplay between the nervous system and the smooth muscle cells, the relationship can be quite complex. Some neurotransmitters may activate the contractile activity of smooth muscles, others may inhibit. The proportion of the two will then dictate whether and to which degree these particular smooth muscle cells are contracted. One of the most important controls over smooth muscle contraction is and pertains to local factors. So local decision making made by the local environment in a form of reflex system. So these local factors, which we mentioned earlier, can include signals from neighboring cells, the pH, the levels of oxygen and carbon dioxide, the osmolarity, and the ionic composition of the extracellular fluid can all, in a combined soup of information, affect smooth muscle tension. Local production of nitric oxide is also very important in inducing the relaxation of smooth muscles. So nitric oxide acts in a paracrine fashion. It's one of these paracrine signals. Certain smooth muscle cells contract in response to being mechanically stretched. They contain mechanically gated ion channels, mostly of the sodium type or the potassium, and once the deformation of the plasma membrane takes place, they then are activated to release calcium and therefore begin the cross-bridge cycle. There are two main classes of smooth muscle cell cooperation. The first type is called the single unit smooth muscles. Here, a stimulus at a single muscle cell, smooth muscle cell, is able to trigger neighboring muscle cells to contract because of the presence of gap junctions. The second class are the multi-unit smooth muscle cells, which contain cells that respond to stimuli independently, and they don't contain any or few gap junctions. The benefit of the former, the single unit combination, is that the autonomic nerve fiber only has to make contact through a number of these varicosities with some cells, and neighboring cells are connected by gap junctions will automatically be activated as a result. Let's turn our attention finally to cardiac muscle. There's only one location in the body where cardiac muscle is found, and that's in the heart. On average, the number of nuclei they contain is between 1 and 2. They do have striations, indicating that they use a coordinated infrastructure within their cytoplasm. But they are branched, and they have intercalated discs at their ends, which contain force-bearing desmosomes, as well as gap junctions for transmitting biochemical information. The gap junctions are critical in coupling the entire heart to coordinate its sequence of contractility. About 1% of the heart muscle consists of nodal cells, and these nodal cells have the ability to synthesize their own action potentials. These will be the pacemaker cells. Another important difference of cardiac cells compared to other muscle cells is that these cardiac cells have an absolute refractory period, the time that they can't respond to a second action potential, of about 250 milliseconds, a quarter of a second. And that prevents the heart from going into tetanus. Tetanus will be lethal to the body if the heart is frozen in a contracted form. The branching nature and the intercalated discs, as well as the location of gap junctions, is nicely illustrated in this single diagram. The mechanism which activates the contraction of cardiac muscle is pretty complex and is discussed at length in the chapter on neurons. The major difference, however, is that the membrane of cardiac muscle cells contains a whole host of different types of proteins with specialized evolutionary adaptations designed to support the physiology and anatomy of the heart. One such evolutionary adaptation is that the refractory period is huge compared to that of skeletal muscle. The anatomical and physiological differences between the different types of muscle in the body are compared in table 9.6 over the next 2 slides.